Hello, everyone, and welcome to today's Steve Berger winner presentations. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is sponsored by Agilent. So let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you'd like during the presentation. To do so, simply type them into the Ask a Question box and click Send. Please address your questions to a specific speaker. We will have answers as many questions if we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, simply click on the Ask a Question box located on the far left of your screen. I now present today's speakers. First, we will have Chandimal Pathmasiri, a PhD candidate at University of Illinois at Chicago, and he will be presenting the topic of investigation of altered phosphoinositides in the neurodegenerative disorder. That's the Niemann-Pick Type C1. Then Dr. James Dodd, postdoc in Professor Aaron Baker's lab at North Carolina State University, presenting combining credentialing at ion mobility spectrometry and tandem MS to detect identity and validate metabolites in untargeted analysis. For complete biography on today's speakers, please visit the Biography tab at the top of your screen. Dr. Chandimal Pathmasiri, you may now begin your presentation. Welcome, sir.
Hello, everybody. My name is Chandimal Pathmasiri from Cologna Lab, Department of Chemistry at University of Illinois at Chicago. So today I'm going to talk about a project that we did to investigate altered phosphoinositides in neurodegenerative disease Niemann-Pick Type C1. So first, let's look at what is Niemann-Pick disease. Niemann-Pick disease is a autosomal recessive neurodegenerative lipid storage disorder. The patients that has these mutations patient that has that disease has mutations in NPC1 or NPC2 genes. So NPC1 and NPC2 genes make proteins that are found in lysosome that involves in lipid transportation. So when cells has these mutations, they are unable to move molecules like cholesterol through the endosomal lysosomal system and create a traffic jam of lipid inside the cell. That led to several downstream consequences like inflammatory response, abnormal signaling, and neuronal cell death. Towards the right side of the slide, what we are looking at here is Calbinden staining. So Calbinden stain for Purkinje neurons. What we see here is a NPC1 mutant mice cerebella. At the early stage of the disease, we see loss of Purkinje neurons from the lobules 2 and 3. And with the progression of the disease, we see that Purkinje neurons starts to die in the other lobules too, and it continues towards the later lobules. So this is known as a pattern neurodegeneration in Niemann-Pick disease type C1. So because this is a lipid storage disorder and has a pattern neurodegeneration, we were interested of looking at changes of distributions in lipids with the progression of the disease. So the best way to do that is mass spectrometric imaging. First, I will walk you through the workflow of mass spectrometric imaging, how we do this analysis. Initially, after the euthanization of animals, we dissect the tissues and flash freeze these tissues on dry ice. So that helps to keep the spatial integrity of the tissue. And then we section this tissue into 10 micron thickness sections onto a MALDI plate and then wash with ammonium formate. So the washing step is optional depending on the analysis. After that, we apply a matrix using sublimation technique, which gives a very uniform coating of matrix. In this analysis, we use matrix named as 9-aminoacridine, which is very good in negative ion mode. In the mass spectrometer, there is a laser that can raster through this tissue, and each of these laser spots creates a mass spectrum. And there are softwares like MSI Reader and Tissue Viewer. They can regenerate heat maps with the data that is created in this analysis. So heat maps shows the distribution and the abundance of each of these lipids in the tissue. We can look at the region of interest, and also there is an algorithm created in our lab recently to look at replicate analysis. So what happens in the algorithm is it filters out noise, and it does peak alignment and normalize the signal between, let's say, mutant and wild type. And it allows us to create an average mass spectrum for all these biological and technical replicates, and allows to look at changes in the abundance of lipid. So we did this analysis for this mouse model at week 3, week 7, and week 9. So these are 3 time points that we study this progression of disease, where week 3 is pre-symptomatic, week 7 is symptomatic, and week 9 is pre-terminal. Week 11 is the average terminal time point of these mutant mice. In this image panel, we have NPC1 wild type in the left side and NPC1 mutant in the right side. And what we are looking at here is the molecule at 1045.5 mOZ. What we observe is this molecule is depleted in the mutant compared to wild type at all 3 time points. So what is this molecule? To find that out, we did on-tissue MS, and looking at these databases for the fragmentation pattern, we identified this lipid as phosphatidyl inositol bisphosphate, or shortly known as PIP2. So PIP2 is a class of, phosphoinositides are a class of lipids, and PIP2 is one of these phosphoinositides. Let's look at what are these phosphoinositide lipids. Phosphoinositides are composed of a diacylglycerol, which is linked to a inositol head group via a linker phosphate. So this is called as phosphatidyl inositol. This molecule can get further phosphorylated to make monophosphorylated, bisphosphorylated, and triphosphorylated phosphoinositides. Not only that, but also, depending on the position of the phosphorylation, there are different positional isomers for monophosphates and bisphosphate. Interestingly, these different positional isomers has different functions in the cell. Even though this class of lipid is very important signal in lipids, it has been very challenging to study these lipids due to several reasons, like the abundance. The abundance of these molecules are very low in the mammalian cell. For monophosphate and bisphosphate, the abundance is even less than 1% from total phospholipid. Also, for PIP3, it is less than 0.1% from total phospholipid. Also, in addition to the abundance, ionization efficiency of these molecules in the mass spectrometer is also very low. So next, I will show you how we overcome these challenges in order to analyze this class of molecules in mass spectrometry. Okay, so up to now, what we know from the mass spectrometric imaging is that PIP2 is depleted in the mutant compared to wild type, but we do not know which isomer is depleted. So in order to find that out and also to study this class of lipid further, we utilize liquid chromatography mass spectrometry. This is the workflow that we follow to do this analysis. First part is extraction of lipid. We use week 3 pre-symptomatic, week 9 pre-terminal mouse cerebella in this study. We lyse the tissue in NP40 buffer, which helps to get the membranes dissolved. And we extracted phosphoinositides using acidified bright dye extraction, and then these extracted lipids were analyzed in 2 major approaches. One is with a derivatization to this molecule, and the other approach is without a derivatization, but using a higher pH mobile phases to help to ionize this molecule in negative ion mode. Especially with this non-derivatized method, which I have shown in the right side with the green color background, we were able to separate the positional isomers of bisphosphates in the reverse space. So next, I will talk a little bit about the TMS derivatization method, show the data from that method, and also go to the data for the non-derivatized method. So TMS derivatization is a very important, very simple reaction which methylate the hydroxide that are connected to the phosphate group. How to do this reaction is to by this commercially available reagent called TMS diazomethane in hexane and incubate at room temperature for 10 minutes. The benefit of the resulting molecule is, major benefit is the higher ionization efficiency in positive ion mode. And also, the resulting molecule has very minimum carryover effect in the liquid chromatography system compared to the reactant. And the stability of this resulting molecule is higher than the reactant. This is data from this derivatized analysis, where for PIP2, we observe the same information as we see in mass spectrometric imaging, where in early and late time points, this lipid is depleted in both, depleted in mutant and mutant at week 3 and week 9. So in addition to that, also we see monophosphates are depleted in mutant at both time points. And when it comes to the PI, it is not significantly different at week 3, but is depleted at week 9. As I mentioned earlier, in the non-derivatized analysis, we were able to separate the positional isomers using reverse phase chromatography, and we see that PI45P2 is depleted in mutant. So PI45P2 is the major PIP2 species in the mammalian cell. So let's look at how these molecules are synthesized in the cell. So this starts in the endoplasmic reticulum, where the CDP diacylglycerol converts into phosphatidyl inositol by linking with a inositol head group. And then these molecules get transported to plasma membrane and Golgi apparatus with the help of PI transfer protein. And at these locations, there are kinases and phosphatases that can synthesize the monophosphates, bisphosphate, and triphosphate. So kinases add a phosphate to the molecule, and phosphatases removes a phosphate from the molecule. PI is the precursor for all these molecules. So with knowing that PI is the precursor for PIP2, we decided to look at the distribution of this PI in the mouse cerebellum. Even though we don't see a significant difference in abundance at early time points, if you look at the distribution of PI and PIP2, they are very different. So PIP2 is in the white matter and PI is in the gray matter. To have a closer look at this, if you look at this overlay image where we see PI in red color and PIP2 in blue color, we see the clear difference. So PI is in gray matter, especially found more in the granular layer, where PIP2 is found in white matter. White matter is rich in myelin. So myelin is very important in this demyelination or neurodegenerative diseases. Myelin is a membrane or a insulating wrapping around axons, which has more than 70% of lipids, and myelin helps to speed up neuronal transmission. And if there is any defects in myelin or any defects in the composition of the myelin, that leads to retarded impulse coordination. When it comes to NPC1 disease, it has been known that demyelination is seen in NPC1 disease. If you look at these images, which are staining from myelin basic protein. So myelin basic protein is a prominent protein in the myelin. We see that this protein is depleted in several regions of the brain, suggesting demyelination is seen in NPC1 disease. So knowing that information, we were interested of looking at the compositional changes in myelin, and we decided to enrich myelin. How we do that is we lyse the tissue in 0.35 molar sucrose, and then we pour this onto a 0.85 molar sucrose and do high-speed centrifugation. And you can see myelin in the interface of these two solutions. What we do is we take this myelin and then we did phosphoinositide analysis. And we see PIP2 and PIP is depleted in mutant from the myelin from the mutant itself. So it's suggesting that there is changes of the composition of lipids in the myelin. To summarize the talk, first I showed you MALDI mass spectrometric imaging reveals PIP2 depletion, and also further analysis using LC-MS, we see alterations of PIP and PI, and we were able to separate PIP2 positional isomers chromatographically in reverse phase using liquid chromatography mass spectrometry, and we see a significant difference in the distribution of PI and PIP2, which led us to analyze myelin, and we found phosphoinositides are altered in the myelin of NPC1 disease. With that, I would like to thank my group members, especially my PI, Dr. Stephanie M. Cologna, and all my funding agencies. And also, I would like to thank Agilent Technologies for giving me this opportunity. Thank you, everybody.
Hey, everybody. My name is James Dodds. I'm a postdoc in Professor Aaron Baker's lab at North Carolina State University in Raleigh. Today, I'll be talking to you about my recent research using ion mobility spectrometry to help detect, identify, and validate metabolites in the credentialing process that's pioneered by Gary Patty's lab. But before we get into that talk, I want to give a shout out to Agilent Technologies, who funded my virtual travel this year to ASMS with the Steve Berger Award. For those of you who don't know, Steve was an employee of Agilent for some odd 26 years in the sales and marketing department, and he also was academic liaison between Agilent and universities. And so when I was notified that I was going to receive this award, I did a little bit of background information on Steve, and I found a really interesting notification from the Lansing State Journal that was published after his death. And there's a quote there that says, "He was an incredible role model for his two sons and others around him who had witnessed his actions. You could ask for a favor and he'd always say yes without hesitation." And I think, maybe now more than ever, it's more important to focus on what we can do to help and support each other in these times. And I think Steve is a great role model for that. So transitioning from Steve's family to my lab family here at NC State, I've been blessed with a great group of coworkers. You can see their pictures on the left, who are interested in a variety of omics-related sciences relating chemical information to phenotypic outcomes. As it pertains to this talk today, my research is mostly focused on small molecule metabolites, and that'll be the focus moving forward. I think when it comes to metabolomics, there are two main challenges that we as a community sort of face, and those both relate to the idea of significant features. And the first challenge is ensuring that features relate back to biological metabolism. And the second challenge is taking that chemical information from just a raw mass to charge value and relating it back to molecular structure. And we can use things like tandem MSMS to help elucidate molecular structure, but it's not always straightforward. And the talk that I'm going to give to you today basically outlines how we're going to try to address both of those challenges. All right, so the way we can first address the challenge of relating features to metabolism is to incorporate stable isotope labels or isotope logs directly into the experimental design. So if you're involved in cell culture, you're supplying energy sources or a nutrient to your system of interest, and that's predominantly consisting of carbon-12, say in glucose. But if you incorporate stable isotopes like heavy labeled glucose directly into the experimental design, that heavy carbon label tracks throughout the glycolytic pathway and other metabolic processes. And so what you get out of that is heavy labeled metabolites that directly come from biology. And really the utility here is to analyze those samples by analytical methods like mass spectrometry that help relate features back to metabolism. So here in the top left portion of your screen, we have a carbon-12 and carbon-13 labeled cell culture. They're grown up in identical environments to ensure that they produce metabolites at the same rate. Then you usually take those two samples and do an extraction and combine them in ratios that make analytical sense for you to perform the analysis. And so most often this is done in a 1-to-1 ratio. So now you have a sample that contains both carbon-12 and carbon-13 labeled isotopes. And the information you get out of that experiment, like you see on the right, is mass spectrometry information where signals are correlated in abundance based on the way you mix them. And so we have a signal at mass 426 and 436, and they have relatively the same intensity within some error. And the key takeaway here is that these two signals come from one metabolite, and their only difference is that carbon label. And basically, doing this process allows us to collect features that are directly related to biology. There's a different variety of ways of performing this experiment. The one I'll be talking to you about through most of this process is the credentialing platform, again by Gary Patty's lab. There's also another one variant called Miracle, and probably the most well-known of these iterations is the isotopic ratio outlier analysis, or IROA platform, spearheaded by Chris Beecher and is now its own company. These methods are a little different in how they perform the extraction and combining of samples, but basically they incorporate heavy isotope labels directly into the experimental design so that you can get features related back to biology. This process really started when Gary and Aaron were at a conference one day, and basically Gary said, hey, I was wondering if we could use ion mobility spectrometry to help separate and characterize some of these challenging metabolites that we're seeing in our E. coli samples. And Aaron said, sounds like a great idea. So Gary got his student Mike Wang to culture some credentialed E. coli. He did the extractions and then sent us the samples at NC State, where we combined them in the various ratios as needed and analyzed them by liquid chromatography ion mobility mass spec, of course using the Agilent 6560 platform. If you're unfamiliar with ion mobility spectrometry, IMS is a gas phase separation technique wherein analytes are separated based on their size and shape in the gas phase. And so this is really useful for annotating chemical isomers in your sample, like the cis and trans fatty acid that you're seeing on the left. They have the same chemical formula, and the only difference there is the orientation of their double bond. The cis fatty acid is more compact compared to the trans fatty acid, which is more extended. And so the cis fatty acid has a much smaller drift time and encounters less collisions in the gas phase compared to the trans fatty acid. We take those raw drift time values and convert them to collision cross-section values using a calibration curve. And then we can search those CCS values against online databases to use as an additional way to annotate our chemical unknowns in these untargeted experiments. And there's been a lot of effort in the community from both academic and industrial partners to standardize the way we collect and report these CCS values. As the publication that most pertains to the talk today is the Collision Cross-Section Compendium, or CCS Compendium, that's published by John McLean's lab at Vanderbilt University and was largely led by my old coworker, Jackie Pakashi. Basically, the CCS Compendium is an online repository of chemical information, and it includes things like compound name, mass to charge ratio, of course, and collision cross-section value, which I use to annotate my chemical unknowns. When I started analyzing these E. coli samples that Mike gave me, it became pretty apparent to me that there's a lot of redundancy in the way you can annotate the isotopic pairs in these experiments. As I mentioned on the previous slides, the first way to do that is to correlate the mass spectral signals based on their relative intensity match. Usually this is done with a 1-to-1 combination, as you see in the top left. Also, I thought it was useful to analyze just the carbon-12 and carbon-13 samples, and so in that middle pane you can see that the presence and absence of each signal in the other respective sample confirms that they are directly related to biology and their isotope pairs. You can also, if you analyze these samples by high-resolution accurate mass instruments like the TOF that we have, you can use their mass defect and the chemical formula at the bottom to not only verify that the only difference there is the carbon-13, but you can also calculate the chemical formula based on the number of carbons associated with the signal. So very useful information for annotating our unknowns. If you happen to analyze these samples by chromatography, of course, you can look at their extracted ion chromatogram traces, and those should be identical. Just based off of the carbon-13. And the key here is that the ion mobility provides you a cool snapshot of these features. So if you look at the bottom right-hand panel, we have a two-dimensional heat map where black color indicates no ion abundance and the red correlates to ion intensity. And so on the left side at mass 131, we see the carbon-12 signal. On the right, 137, we see carbon-13. And you can see that both those signals have the same centroided drift time at about 16 milliseconds. And that's because that heavy carbon isotope doesn't really affect the size of the molecule, it just affects the mass. And so we take those raw drift time values and convert them to collision cross-section using that calibration curve. And when we search the CCS value of 128 angstroms against the CCS compendium along with the chemical formula, it's pretty straightforward to get a match to N-acetylputrescine with an exact CCS match. I should say that here we do always use MSMS to confirm our chemical annotations. But in this case, I was able to match it just based on CCS. Ion mobility is also very useful when it comes to deconvoluting potentially crowded regions of your chromatographic separation. So here in the top left, we have a base peak chromatogram for the helix separation in ESI negative mode. At that retention time window selection, you can see that we have several mass spectra features, and you can tell that there are several isotopic pairs based just on relative intensity, such as in the blue and green color. However, using the ion mobility is extremely helpful when you analyze those features of low abundance. So you can see there for the feature in red or pink that we have a low abundant feature. It's kind of hard to link those two signals based just on the mass measurement and ion abundance. But when you look at that separation in the mobility dimension, you can see that drift time correlation there horizontally. And it's very straightforward to annotate these or link those signals together. And the converse aspect of that is on the right side, where we have a lone signal that has no abundance match in the mass dimension, nor does it have a partner in the horizontal mobility dimension. And so we don't need to worry about this feature going forward because it probably arises from background and it's not related to biology. The mobility alignment feature of ion mobility in this isotope log workflow is also very useful when analyzing your MSMS data. This slide is pretty crowded, so I'll walk you through it. But basically, during our LC separation, we had the dipeptide Val-Glu and glutamate co-elute in our helix separation. This isn't really surprising because these two molecules are very similar structurally. And so they co-elute, but you can see that the dipeptide is much larger than the glutamate ion. So it elutes from the drift tube later compared to the smaller glutamate ion. And this is useful because the glutamate precursor is actually a product ion of activation for Val-Glu. And so when you just analyze these samples by LC-MS, it's kind of hard to deconvolute that chimeric spectra. But with the mobility separation, L-glutamate is much smaller, so it's readily resolvable from the dipeptide. I should say that for glutamate, I was able to annotate this compound again based on just the chemical formula and CCS. You can see the match to the literature there on the left side of your screen. But also what was unique here is that there are two other isomers in the CCS compendium, N-methyl aspartate and no-acetylserine, and those compounds are much smaller in their CCS compared to glutamic acid, so it was a pretty straightforward annotation there. I want to close off my talk with a cool story about the utility of both ion mobility and chromatography. So when I was analyzing the E. coli extracts, I noticed a bimodal distribution for the mass charge ratio 565, and I saw two peaks at retention time 3.8 and 4.0, and there's a good bit of, there's decent separation there in the LC dimension. So I pulled the ion mobility spectra out of each of those respective peaks, and you can see that the early eluter has a collision cross-section of 207, and the later eluter is a little bit larger at about 208, 209 square angstroms. This is a small difference in CCS, but it was reproducible and is definitely there. When I looked at that chemical formula and those CCSs and the compendium, I got very accurate matches to UDP glucose and UDP galactose, the two stereoisomers. And so I was pretty confident that there were two isomers in this sample. The challenge here is that both these stereoisomers, unsurprisingly, have the same MS-MS spectra, which you can see from Metlin there on the right, so I can't use that to differentiate them. Thankfully, Sigma Aldrich, or I should say Millipore Sigma now, bailed me out on this because they actually have standards for both UDP glucose and UDP galactose, and matching those two compounds with the standards was very straightforward in subsequent analysis. So I thought this was really useful because I wouldn't have known that there were two isomers there without the chromatographic separation, but I wouldn't have been able to annotate them without the ion mobility. So really cool example there of this technology. So in conclusion, I think that the isotopologs, workflows, and ion mobility are a natural fit. They have a lot of experimental behaviors that make them uniquely suited for each other. And of course, you need libraries and standards to validate these annotations. I don't think that's going to change anytime soon. For future applications of this work, I think that timescale experiments or flux analysis or even tracer work could be very useful, and ion mobility helps provide you a snapshot of those credentialed or those verified isotopic pairs. I'd like to thank my lab members for all their help on this talk, our collaborators, Gary and Mike at Washington University, and of course, I'd like to thank Agilent Technologies for for supporting our research there in the Baker Lab, and in particular here with the Steve Berger Award. Those are the other sources of funding I have, and I'd be glad to take any questions you may have. Hey, can you hear me, Antonio? All right, so it seems like Susie's having a little bit of technical difficulties here on her end. I don't know if I can see any of the question and answer comments from my screen, but I will say one of the things that I get asked most often about this work is if the credentialing process can also apply, or excuse me, the ion mobility verification part can also apply to IROA and the Miracle platforms. And there's also another lipid isotopolog workflow as well. And the answer is absolutely yes to all those questions. The method is the same. It's just annotating isotopes by ion mobility. So the managed Q&A session is at the very bottom of my screen. So I see that. Oh, okay. Oh, I see. Okay, for the questions coming through. So for Chandimal, one of the people in the audience wants to know what kind of MALDI-TOF you're using.
Yeah, so the instrument that we use for these experiments is SciX MALDI-TOF 4800 system. And also one other important thing is this instrument has a laser with a diameter of 50 micrometers. So that decides the image resolution of the analysis. So the image resolution of our data is 50 micrometers.
Perfect.
Thank you, gentlemen. I apologize for my little technical problem there. So this next question is for you, James. You showed in your talk that ion mobility can help provide some ways to link the isotopic pairs for the credentialing process. Does this work for ROA and Miracle as well?
Yeah, absolutely. They all function very similarly in terms of how they generate their isotopologged metabolites. There's just various slight nuances in how they combine those extracts. So ion mobility is extremely useful, and it doesn't matter whether you use like FAMES or trap ion mobility or traveling wave. All of those platforms will do very similar analyses for this. And really the challenge is the data analysis right now, which is currently manual, but that's a good question.
Thank you. And I want to thank our audience members for these great questions coming in. Chandimal, you showed PI, PIP, and PIP data for the fatty acid composition. Have you looked at other fatty acid composition for these molecules?
Yeah, so in this analysis, we do see several fatty acid compositions for PI, PIP, and for PIP2. The data that I have shown is with the fatty acid composition of 38 carbon and 4 double bonds, which is one fatty acid is 20 carbon with 4 double bonds, and the other fatty acid is 18 carbons. So this composition is the most abundant composition in the mammalian cell. So that's why I showed that one. But we do have data for other compositions as well. And from those data, what we observe is the same observation, where the PIP2 and PIP is depleted in week 3 and week 9 NPC1 cerebellar. So yeah, it is important to look at different fatty acid. If the fatty acid chain length is different and if they are showing different trends, that may be meaningful for this study. But in this case, I would say the changes that we see is related to the head group. So it does not depend on the composition of the fatty acid. Yeah, thanks.
Thank you. And I do want to remind our audience members that if you have any questions you'd like to ask, just do so. Simply click on that Ask a Question box located on the far left of your screen. Okay, our next question is for you, James. The mass and scamp, is that it? Equation?
Yeah, close enough.
Says that the heavy labeled isotopes should have a larger CCS drift time compared to the light metabolites. Why do they have the same drift time in your results?
Yeah, so by first principles of kinetic theory and the Mason-Champ equation, basically although the shape and size of each of the C12 and C13 labeled molecules is the same, the mass is actually a component of that equation. And so since the mass of the carbon-13 isotopes is a bit more, the CCS should be larger, and then the drift time itself should also be larger as well. That is true if you look at the centroids of the drift time for the C12 and C13 isotopes. Generally speaking, the C13 one is a bit larger, but the difference we're talking about here is approaching the area of reproducibility of our instrument, and you need much more higher resolving power to be able to really distinguish that every single time. So you need something like the SLIM or the mobile ion to sort of get into that realm.
Thank you. And Chandimal, can you comment on the amount of material needed for this phosphoinositide analysis?
Yeah, so in this study, I did several analyses. One is from the mouse cerebellar extract, and other analysis is for the myelin. So also I used 2 instruments. So in terms of instruments, so I use Agilent 6545 instrument, and also I use Agilent 6550, so which has a higher sensitivity. So for the 6545, when I do this analysis in that instrument, I usually start with at least 150 micrograms of protein equivalent. So I normalize always with the protein composition, and this is for the brain tissues. So take 150 micrograms of protein equivalent at least from the samples, and then do the lipid extraction, derivatize, and then I inject in the 6545, I inject in between 10 to 20 micrograms, and that gives a very good signal. And so when I did the myelin isolation, so because I did the myelin isolation from a cerebellum, especially at 3 weeks, I do not get much of material. So what I did is I did this analysis with 50 microgram protein equivalent. Also, I have done this analysis for cells using 50 micrograms protein equivalents using 6550 instrument. And so I extract the lipids out of 50 microgram protein equivalent and inject 0.5 to 1 micrograms. That gives a very good signal. But I do want to mention when doing this analysis, one very important thing is this derivatization reaction. And it heavily depends on the quality of the reagent. I do have seen sometimes the reagent does not work if you keep it outside for a long time. So I would recommend if you want to do this experiment, always keep this reagent in 4C and check the derivatization reaction using standards. Yeah, thank you.
Thank you so much, Chandimal. And James, this next question is a two-part question. Do you find any issues with incomplete incorporation of C13 during culturing? And can you detect this in your experimental platform?
So to be honest, this isn't something I've dived into too deep. And I don't know if the way you evaluate that is if you look at where the C13 peak is in the mass spectra and then look sort of, well, it'd be to the left on the mass scale. If there's not complete incorporation of the C13 tag. If that's so, maybe a little bit, but not much. I see that, and I don't know if there's any way to get feedback on if this is correct, but I think the question's coming from Fernando Tobias at Ohio State. If so, and I can send you an email after this following up and clarifying what you mean, and then I can go back and look at the data. But I think you would evaluate that on the left side of the C13 peak. It seems to be pretty good for me. Again, I didn't do the extraction, so I can't say anything about that part, but I can definitely send you a private email after this is over, and you can sort of let me know what you're thinking.
Thanks, James. And here's another one for you, actually. Would the traveling wave ion mobility and trapped ion mobility instruments provide similar results with a credentialed metabolite?
Yeah, they should do exactly the same thing in terms of their alignment of those isotopologues. With the newer traveling wave and the trapped ion mobility, you'll get more resolving power. So you might start to see the C13 peak grow a little bit, quote unquote, in CCS relative to the C12. But as far as feature finding and things like that go, it should still be very straightforward.
Thank you. And we have time for a couple more questions. Chandimal, have you considered doing IMS to support the differentiation of isomers of PIP2?
Oh, that's a great question. Yeah, we have tried. Also, I think this is something that I would like to talk to James too, because he has been working on IMS. Yeah, so it gives a good separation for PIP2 actually. But monophosphate is the ones that we are having troubles to separate because it has only one phosphate and only the position is different. But yeah, I would like to work more on IMS in order to try to separate this in IMS. Thanks for the question, Fernando Tobias.
Yeah. To that point, Chandimal,
yeah.
Oh, I'm getting a lot of feedback there. Yeah, at NC State, we have a collaboration with Dave Muddyman, and I saw that you had looked at the MSI platform to do your data analysis. And so we used Dave Muddyman's MALDI-MSI, IR MALDI-MSI source on the Agilent 6560 in a very crude setup. And it does work. You have to sync the laser pulse with the trap release time in the drift tube, and it can work. And I think there's definitely some room there for to get that going. I think Bruker is also trying to get the timsTOF to have their MALDI source on the front, and I think that's a good thing to look into too. So you might be interested in that work. Yeah,
definitely. Yeah, thanks. I will look into that more.
Thank you both. And we have time for one more question. This last question from an audience member it's not directed to either one of you, so one of you can choose to answer. What model of MALDI-TOF was used?
Oh, that's for my experiment. I use AB-SciX MALDI-TOF 4800 system.
Thank you for that. And thank you both for your time today and for your important research. Would either of you like to have any closing remarks before we close today?
You can go first if you want.
Yeah, go ahead.
Oh, okay. No, I think it's been a very good talk, and I think that the key, at least from what I saw of both of our talks, is it's very hard to pinpoint molecular structures due to a lot of isomers. And I think that, you know, MS and ion mobility in conjunction with chromatography is a very good way to go.
Yeah, from this, especially from this ASMS, I learned a lot of new advances in the field that can be used for molecular identification. And it has been very helpful having this conference in online format. So I think mass spectrometric recent advances are very useful in terms of biological analysis to answer biological questions. And also, I would like to thank Agilent for especially arranging this virtual event and giving us the opportunity to present our work and get feedback. It has been very helpful, those questions and everything. Thank you very much.
Absolutely.
And I want to thank you both again for this presentation and for your time. Before we go, I'd like to thank the audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand through June of 2021. Until next time, be safe, take care, bye-bye.